All right, I think we are jumping right into the tumorigenesis session. To start the session off, I have the pleasure of introducing Dr. Christine Pertillis from Johns Hopkins University, and the title of her talk is Novel Therapeutic Strategies for MPNST.
Okay, thank you, Angie. Can you all hear me? Okay, great. And I think if I understand correctly, I now have the ability to control the slide advancement. Can you see my slides okay? Great. So I'd like to thank the organizers for the opportunity to share some of my lab's work today. And I'm going to be talking about our preclinical work investigating the effects of several novel small molecule compounds in models of MPNST. Double click on the slide. Okay. Great, thank you. Okay, so these are my disclosures. And so I'll start, just my lab's interest is really focused on cancers with dysregulated ERK signaling. And of course, as you know, ERK is one member of the MAP kinase family of mitogenic signaling molecules, and it's responsible for multiple cellular processes in both normal and cancer cells. And it turns out that ERK signaling is hyperactivated or dysregulated in approximately 30% of human cancers. I am not advancing, I'm so sorry. How's that? Okay. And typically ERK signaling hyperactivation or dysregulation is a result of mutations in RAS or BRAF as shown here. And these findings have suggested for now more than 20 years that the pathway might be a target for drug development. And so MEK inhibitors were therefore developed as a strategy to address what was considered at the time high phospho-ERK cancers. And in fact, this was prior to the discovery of BRAF mutations in cancer. And these compounds, as a class, have the unique advantage of being among the most selective of clinically available small molecule inhibitors. And the first of these to enter clinical trials was CI-1040, but unfortunately, it really didn't demonstrate meaningful clinical activity in the first human clinical trials. But following the identification of BRAF mutations in cancer, our lab was among the first to demonstrate that BRAF mutant cancers are in fact selectively sensitive to MEK inhibition, and we did that using the compound PD325901. Following these discoveries, that led to the development of several other in-class compounds, including those shown here, several of which have now become FDA approved for the treatment of cancer. So as we think about the classes of tumors in which MEK inhibitors might have a role, tumors with loss of NF1 emerge as another category that might predict ERK dependency and therefore sensitivity to MEK inhibition. And so, of course, loss of NF1 somatically occurs in multiple tumor types, including melanoma, glioma, gastrointestinal stromal tumors, non-small cell lung cancer, rhabdomyosarcoma, and malignant peripheral nerve sheath tumors. And so we were really interested in melanoma at the time and looked at the class of melanomas that didn't have mutations in BRAF or mutations in NRAS, and that's a smaller subset, but it turned out that a large percentage of that category, in fact, had loss of function mutations in NF1, and we went on to show that the tumors with loss of NF1, melanoma tumors with loss of NF1, were also associated with RAS activation, MEK dependence, and sensitivity to MEK inhibition. And so, NF1, of course, is a RAS GTPase activating protein, or a RAS gap, which means that its biochemical role is in the conversion of GTP-bound active RAS to GDP-bound inactive RAS. And therefore, its loss in cancer is associated with increase in RAS GTP and RAS effector signaling. And MPNST, or malignant peripheral nerve sheath tumor, is a cancer that really needs no introduction to this audience, is a high-grade malignant soft tissue sarcoma that arises frequently in patients with NF1. And it is genomically characterized by loss of NF1, activation of multiple receptor tyrosine kinases, loss of CDKN2A, and therefore activation of the cyclin D CDK4 complex activity, and mutations in EED and SUS12, members of the polycomb repressor complex. And several groups had previously examined the potential role for MEK inhibition in MPNST tumor models. And while some activity was noted, the effects overall really suggested a partial response, as shown here in this figure from Nancy Ratner's group using tumor xenografts, and also in this paper by Rebecca Dodd, which used genetically engineered mouse models of NF1 deficient sarcomas that develop MPNST. And in fact, these effects here again shown in Rebecca's paper using PD325901 in MPNST models, demonstrating that the effects of MEK inhibition on tumor growth inhibition were partial. And so we posited that ERK is hyperactivated in tumors with loss of NF1 and hyperactivation of RAS, but that MEK inhibitors are only partially effective. And we therefore hypothesized that identification of the adaptive changes in ERK signaling upon MEK inhibition would identify targets for combination therapy. And so the work that I'll show you in the next several slides is work done by a fantastic postdoc, Jaiwan or Ava Wong, who joined my lab several years ago. And so Ava began with a large panel of available MPNST cell lines, as well as several that we had developed in our laboratory. And she found that, she characterized these lines for expression of NF1, P16, and SUS12. And I will just mention here that at Johns Hopkins, we manage a large and growing NF1 biospecimen repository through which we bank tumors and other tissues from patients with NF1. We attempt to generate cell lines and PDX and to share both the tissues and the genomic data on these samples through the NF data portal. So if you are interested in samples or cell lines or genomic data, you can find more information about the NF1 biobank using the QR code shown on this screen. So Ava examined the sensitivity of a large panel of MPNST cell lines to MEK inhibition. And she overall found that they're partially sensitive, but that the initial downregulation of phospho-ERK upon MEK inhibitor is transient, with a rebound to a new steady state over time in just a matter of hours. And so as one method to identify the adaptive changes that happen in the short term, we used a phospho-RTK, a receptor tyrosine kinase arrays, and we found that multiple receptors were indeed activated in just 24 hours, including those shown here, such as PDGFR-beta, EGFR, IGF-1R, and MET, consistent with the finding of RTK activation upon MEK inhibition. And so what's important to note is that multiple RTKs, not a single consistent RTK, was activated, and that in these cell lines, we didn't really have a clue from their genomic makeup which RTK or RTKs would become adaptively upregulated. And so we also performed an unbiased global phosphotyrosine kinase, serine threonine kinase peptide array screen, which identified a subset of kinases upregulated by MEK inhibitors at 24 hours in a panel of MPNST cell lines. And included here among the most significantly upregulated kinases shown in this volcano plot are PDGFR-beta, EGFR, and MET, consistent with what we found using our phospho-RTK arrays in the lab. And so our working model is such that ERK is hyperactivated in tumors with loss of NF1, which through negative feedback maintains a low steady state at the level of RTKs and RAS scaffold proteins that regulate RAS activity. But upon MEK inhibition, there's loss of the negative feedback signaling, prompting upregulation of RTKs in general, as well as the scaffold and regulator proteins that regulate RAS activation. So we thought about several potential strategies to overcome these adaptive signals, including inhibition of RTKs or inhibition of additional RAS effector pathways that also become adaptively upregulated, including PI3 kinase and mTOR signaling. And the first, inhibition of RTKs, may have some clinical utility, but perhaps in a more limited way. For example, using the ST8814 MPNST parental cell line, we generate cell lines with acquired resistance through continuous exposure of cell lines to MEK inhibitor over a course of about 5 months, and we found overexpression of MET and HGF in these cells. And Ava demonstrated that these cells then require MET for sustained growth, and they're sensitive to the combination of MEK and MET inhibition. And then we took this combination back to a number of native MPNS T cell lines, and we saw some additive effects of the combination using trametinib plus capmatinib in both cell line and xenograft models of MPNS T. But our findings suggested that the application of RTK combination therapy may be met with several challenges, including the heterogeneity of genomics of MPNS T and our inability to predict which RTKs would be activated in any given model or really any patient. And another challenge is that many of the clinically available TKIs are relatively non-selective and they target more than one RTK, which really makes it unclear how to best choose inhibitors to give in combination with MEK. So we then therefore turned our attention to a novel class of compounds, the SHP2 inhibitors. And so SHP2 is a component of a complex of proteins required to recruit and activate RAS upon signaling and activation from an upstream RTK. And as such, it could represent a central node of convergence for multiple RTKs, which actually eliminates the need to know or predict which RTKs matter in any given MPNST. And several of these compounds are now in early phase clinical trials. I'm showing here data from Novartis using their tool compound SHP099, demonstrating selectivity for tumors with RTK activation. So some of these have mutations or overexpression of specific RTKs. So the mechanism of these compounds, the SHP2 inhibitors, is that they bind to SHP2 in the closed inactive conformation, preventing the association of SHP2 with the cytoplasmic domain of the receptor tyrosine kinase, and then preventing assembly of the complex that's required to recruit RAS to the membrane and thereby activate its effector pathways. So we used SHP2 inhibitors and tested their activity in models of MPNST. Ava first demonstrated that SHP2 phosphorylation and therefore its activation is in fact upregulated upon MEK inhibition, suggesting that it might play a role in combination therapy with MEK. And we found that SHP2 inhibitors themselves also reduce ERK signaling in MPNST cell lines, but like the MEK inhibitors, the effects are transient, with rebound in the first 24 hours. And that the proliferative effects of single-agent SHP2 inhibitors, at least using SHP099, were modest at best. Given together with the MEK inhibitor, however, the SHP2 inhibitor mitigated the upregulation of RAS-GTP that we saw with single-agent MEK inhibitor and led to more sustained inhibition of phospho-ERK and downstream signaling, which in turn led to additive cell growth inhibition, shown here in 10 different MPNST cell lines, the majority of which are really quite sensitive to the combination in vitro. We then tested this combination in 2 animal models using NF90.8 and our own patient-derived xenograft line, JH2002, and we found significant additive effects of MEK plus SHP2 inhibition in vivo, accompanied by profound reduction in Ki67, as well as phospho-ERK and DUSP6 as a biomarker of ERK transcriptional output. And so recently, our lab has been collaborating with several other groups, including Angie Herby's lab at WashU, the Larga Spada and Wood Labs at Minnesota, and Sarah Goslin's group at Pacific Northwest National Labs. And our goals in a project that is now funded by the NF Research Initiative have been to develop a diverse set of genomically characterized MPNST PDX and to test rationally designed combinations, and then to use RNA and DNA genomic data to interrogate and define predictive biomarkers. And so through this work, we further expanded our testing of the SHP2 inhibitor and several other combinations. And so we started these experiments, we actually switched from the tool compound or the preclinical compound SHP99 to the compound that's currently in clinical trials, TNO155. And as shown here, this is the combination of TNO155 together with trametinib in 4 different MPNST PDX models, several of which were developed at WashU in Dr. Herby's lab. One of these models is a JH-derived PDX models. And we were actually a little surprised to see how active TNO was as a single agent in some of these MPNST PDX. And so we went back to the cell lines, and Ava went ahead and she knocked down PTPN11, the gene encoding SHP2, and found that PTPN11 knockdown alone is actually capable of inducing significant growth inhibition. And so we wondered, given the single agent activity of TNO155 and the effectiveness of PTPN11 knockdown in these cells, whether there actually may be other drugs that would synergize with SHP2 inhibitors, perhaps even more potently than the MEK inhibitor. And non-MEK inhibitor therapy might be an attractive strategy, not only because some patients with MPNST will have already had a MEK inhibitor as treatment for their plexiform neurofibroma, but also because SHP2 inhibitors and MEK inhibitors both lead to strong inhibition of phospho-ERK. And therefore, we expect that the toxicities of MEK inhibitor and SHP2 inhibitor would be overlapping and therefore potentially poorly tolerated. And so in looking for other targets to combine with SHP2 inhibition, we were intrigued by results from Don Quell's group, shown here, who identified upregulation of RABL6A and the finding that MPNST were sensitive to CDK4 inhibition with palbociclib. And so CDK4 inhibitors are also an appealing target, since lots of CDKN2A leads to upregulation of cyclin D and the CDK4 complex activity. So we proceeded to test the combination of SHP2 and CDK4/6 inhibition using TNO155 and ribociclib. And we looked at all three pairwise combinations, as I'm showing you here, both the combination of SHP2, MEK, and CDK4/6. And using the combination of SHP2 and ribociclib, we found again that these drugs produce additive effects in vitro in multiple MPNST cell lines. And then we went ahead and designed our in vivo experiments so that we could directly compare the effects of the three pairwise combinations using MEK inhibitor, SHP2 inhibitor, and CDK4/6 inhibitor, as shown here in one of the MPNST PDX models, WU545. And so some of these data are somewhat preliminary and they are unpublished, with further experiments ongoing in an expanded number of PDX models. But so far what we've seen is that the combination of SHP2 and CDK4 inhibition seems to exert the most profound anti-tumor activity of these three pairwise combinations. And so the TNO-ribo combination is a combination that's currently being tested in a phase 2 study in adults with solid tumors, with a currently ongoing focus in a lung cancer expansion cohort. And so based on dosing and toxicity data that come from that study, our hope is to translate this combination of TNO155 and ribociclib into a clinical trial for patients with MPNST. And so I'll just conclude with a couple of wrap-up thoughts here, which is that our data suggests that the limited responses to MEK inhibitor are really largely driven by release of negative feedback and adaptive upregulation of receptor tyrosine kinases, activation of RAS, and then consequent reactivation of ERK and parallel pathways, and that SHP2 inhibitors with MEK inhibitors might therefore mitigate the adaptive upregulation of RAS activity and thereby produce additive effects in vitro and in vivo. And finally, in the unpublished work that I've shown you, SHP2 inhibitor combinations with other small molecule inhibitors, specifically CDK4, may be an effective path forward for the design of clinical trial for patients with MPNST. And so we're currently exploring the possibility of a SHP2 CDK4 combination trial in patients with MPNST. So I'll wrap up there, and I just would like to take a moment to acknowledge the members of my lab that have done the work I've shown you today, specifically Ava Wong, who's responsible for the majority of the in vitro work, Kai Pollard, who runs the NF1 biospecimen repository out of my laboratory, Anna Calizzo, who is responsible for the in vivo work with both the MEK SHP2 as well as SHP2 CDK4 in vivo combinations. I'd also like to acknowledge members of the JH neurofibromatosis team, my collaborators at WashU, Angie Herby's lab, Minnesota, David Largaspada and his lab, several members at Sage Bionetworks, as well as Sarah Goslin at Pacific Northwest, and then Novartis for supplying some funding and the compounds for for the work that I've shown you. And I'll be happy to take questions.
That was a great talk, Christine.
Thank you.
Peeking on Slido to see what questions come through, but I will start things off with a question about one of the discussions we've had in the past. But single drugs alone are unlikely to be a home run for these tumors, and with combinations, we always run into toxicity issues. How many drugs do you think we're going to be able to combine? Because even theoretically, even 2-drug therapy, eventually patients are going to develop resistance to.
Yeah. Definitely true, and great points as we think about how we move some of these small molecule inhibitors into clinical trials in people. There's definitely a precedent for the idea that if we give single agent small molecule inhibitors, the tumors will ultimately figure out ways to evade them and develop acquired resistance. And so certainly one of our goals is to understand how tumors with NF1 loss acquire resistance to SHP2 inhibition. So we'll be eager to understand that a little bit further. And then as we combine drugs, I think what's really important is thinking about how we can design combinations that limit the overlapping toxicities, right? And so that may be one limiting factor in moving forward with SHP2 MEK, even though we were pretty excited about the preclinical activity of that combination. The fact that both agents are causing pretty profound downregulation of phospho-ERK, and that leads to a very sort of well-described known set of side effects, we really may not be able in people to safely combine SHP2 and MEK inhibitors. And so thinking about drugs with non-overlapping toxicities is really critical here. That's one of the reasons we're more excited about how we might move forward with SHP2 CDK4 combinations. Whether we can combine 3 safely, I think really remains to be seen.
Let's see. Someone came through with a question. Are there any known mechanisms to SHP2 inhibitors, CDK4/6 pathway, a resistance mechanism, or an adaptation?
Yeah, so some of the groups, there have been groups, in fact, the Novartis group has described resistance to SHP2 inhibition in the form of upregulation of receptor tyrosine kinases, specifically FGFR4. But I think as far as NF and PNST, we don't yet know, but that is an area that my lab is investigating.
Someone else came through, very nice talk. TNO alone seems to be fairly potent. Can you speculate on further increasing the TNO dose to see a more profound effect?
Yeah, that's a great question, actually. So like I showed you, TNO155 was more potent as a single agent than what we had seen when we were using the tool compound SHP99, right? And so we had initially brought in SHP99 as a way to overcome and mitigate the adaptive upregulation of receptors and RAS activity seen with MEK, thinking that MEK was sort of the key compound and then SHP was coming in as a way to sort of overcome adaptation. But as we think about the SHP2 inhibitor as the more potent drug itself, we have done experiments in our mouse models to push the dose up. So the majority of experiments that I showed you in vivo are using a 7.5 mg per kg dose. When we double that and we go to what would be thought of as a single agent dosing of 15 mg per kg, we actually didn't see much difference. So we may have capped out on SHP2 inhibition molecularly, and we didn't really see any added benefit of doubling the dose. So I think we'll just have to see what those side effects look like as we push higher and what that might look like in people.
Thank you so much, Christine. That was a wonderful talk. I will turn it over to Dr. Chi to introduce the next speaker.
Okay, so our next talk is by Dr. Lawrence Paco. She'll be talking about a case-only study in 1,333 neurofibromatosis type 1 patients to identify genetic modifiers of cutaneous, subcutaneous, and plexiform neurofibromas. Welcome, Lawrence.
Hello to everyone. I'd like to thank the organizers of these conferences for giving me the opportunity to present here to you the results of our study in the identification of genetic modifiers in the development of neurofibromas in a French cohort of NF1 patients. As we all know, neurofibromatosis type 1 is a genetic disease with autosomal dominant transmission for which more than 95% of patients carry a pathogenic loss and function variants in the NF1 gene. The disease is characterized by complete penetrance, pleiotropic manifestations, and an expression that can vary greatly between individuals. NF1 patients carry heterozygous mutation of NF1 in some or all of their cells. At some point in their development, some specific cells will inactivate the wild-type allele of the gene, leading to an uncontrolled proliferation and to the growth of benign tumor, as is the case for neurofibromas, which result from the overproliferation of Schwann cells. If these Schwann cells accumulate additional genetic events, such as mutations in TP53, they can be at the origin of malignant peripheral nerve sheath tumors. Only a few NF1 pathogenic variants have been correlated to a specific presentation of the disease. They are summarized on this image, representing neurofibromin with its main functional domains and variants that are reported in the CBO portal database, especially variants leading to the deletion of the methionine at position 992, or missense variants affecting methionine at position 1149, or adenine at position 1809, associated with the complete absence of neurofibromas. On the contrary, missense variants affected codons 844 to 848, arginine at position 1276, or lysine at position 1423 are associated with an increased prevalence of either plexiform or spinal neurofibromas. Complete deletions of the gene cause particularly severe expression of the disease, with dysmorphism, cardiovascular malformations, learning disabilities, and a high burden of neurofibromas. Except for the specific variants, the nature of NF1 variants was shown to have only a low influence on the phenotypic variability of the disease. Intrafamilial phenotypic correlations and animal models suggest that part of the variable expressivity of NF1 could be explained by modified genes independent of NF1. This is especially the case for cafe-au-lait spots and neurofibromas, for which the additive genetic component is estimated to account for 20 to 30% of the total phenotypic variability. In order to identify these genetic modifiers, many candidate gene approaches have been successful in the past. I will now present to you the results of the genome-wide association study that we conducted in our large French cohort of NF1 patients. For this work, we will try to identify the genetic modifiers implicated in the variable expressivity of cutaneous, subcutaneous, and plexiform neurofibromas in NF1 patients. For this work, we benefited from the vast recruitment of NF1 patients in the NF France cohort. These patients were enrolled through 3 clinical research programs between 2002 and 2013 and represented more than 1,500 affected individuals from more than 500 families. Patients were clinically evaluated using a standardized questionnaire written by the Neurofibromatosis French National Reference Center. They were all sequenced for the NF1 gene at the Cochat Hospital in Paris using a high-throughput sequencing technology. Cohort selection started by the sequencing of the NF1 gene. We excluded from the study patients without an NF1 pathogenic variant or with one of the NF1 variants that are associated with a specific form of the disease. The steps of whole genome genotyping, imputation of additional variants, and the application of quality features led to the exclusion of patients with low-quality genotyping and of low-quality genomic variants. A total of 1,333 NF1 patients, genotyped for more than 7 million common genomic variants, were included in the genome-wide association study. We then split this cohort into a discovery and a replication samples, and on both groups, Dr. Audrey Sabac performed the association study for the different types of neurofibromas by using a mixed model including 4 covariables. I then performed the fine mapping and prioritization steps to identify the genomic regions associated with the different traits and the candidate modified genes in the outcome of cutaneous, subcutaneous, and plexiform neurofibromas. Here is the global representation of the association study performed for the 3 main types of neurofibromas in the discovery sample alone. On these Manhattan plots, each dot corresponds to 1 genomic variant. These variants are plotted according to their genomic location on the x-axis and to their association with the trait on the y-axis. The red horizontal line corresponds to the genome-wide significance threshold, that is usually applied in genome-wide association studies. The blue horizontal line corresponds to the threshold that we applied to select regions that were suggestive of an association with the threat, though not reaching the genome-wide significance threshold. Now, let's give an example and take a close look at this highly significant region on chromosome 9 in the study of plexiform neurofibromas. So here is a zoom of our candidate region. Variants are still plotted according to the genomic location on the x-axis and to their association with the threat on the y-axis. The lead SNP, here in purple, is the genomic variance with the lowest p-value in the region. It is the reference for the fine mapping of the region. The other variants of the region are colored according to their linkage disequilibrium with the lead SNP. They all constitute variants that may be implicated in the development of plexiform neurofibromas. We observe that in this region, 60 variants have a p-value under the genome-wide significance threshold. Annotation of these variants allowed us to point 4 candidate protein coding genes associated with this genomic region. We performed similarly in other threads and looked at the regions that suggested of an association with plexiform cutaneous or subcutaneous neurofibromas with a p-value for the lead SNP under the threshold of 10⁻⁶. Altogether, we identified 12, 3, and 4 regions suggestive of an association with plexiform cutaneous and subcutaneous neurofibromas, respectively. These regions were associated with several protein coding and non-candidate genes that might be implicated in the outcome of these neurofibromas. The second part of the study consisted in the identification of genomic regions that showed evidence of replication in the replication sample. So for this, we looked at regions that were suggestive of an association in the discovery sample at the 10⁻⁵ threshold, and considered them as replicated if they included at least one variant that was significantly associated with the threat at a 5% threshold in the replication sample. So here's the representation of our replicated regions as Miami plots. So you can see at the top of the plots the Manhattan plots in the discovery sample for each thread, and at the bottom the mirror Manhattan plots in the replication sample. Replicated regions are spotted in red. Well, I can't go too much into details on this analysis, but altogether we identified 4, 2, and 6 replicated genomic regions, respectively associated with plexiform cutaneous and subcutaneous neurofibromas. Like previously, the annotation process pointed out several candidate modified genes that might play a role in the development of these neurofibromas. To conclude this work, we were able to identify several regions that show evidence of an association with either cutaneous, subcutaneous, or plexiform neurofibromas. Especially, these regions are different between the distinct phenotypes that we studied. This underlines the existence of distinct pathophysiological processes at the origin of these neurofibromas. We can note that several candidate modified genes are implicated in pathways such as the Ras pathway, the cell cycle, or the myelination process of Schwann cells, which are the main cells implicated in the emergence of neurofibromas. In conclusion, our study confirms the existence of modifier genes and points out different candidates. I'd like to thank my research team and other groups that we have worked with for their implication in this project. I give a special thanks to the NF France network for the fantastic recruitment of the French cohort. And of course, I thank all the patients who put their trust in us, and I thank you for your attention.
That was a great talk. Now we're open for questions. We have a few minutes. Maybe I'll lead by asking you. So of all of the sort of SNP candidates, variants, are there any shared ones across the 3 different types of neurofibromas?
In fact, no, that's what was really surprising. At first, there is really no common region between the different type of neurofibromas. So clearly there is something different that's happening and that's at the origin of the different types of neurofibromas. And that's what was already suggested in the literature. So in the end, we're not so surprised about the result.
While we're waiting for other questions coming, I have another question. Can you speculate or shed some lights on, how these variant regions or SNPs might influence the downstream genes? Are they augmenting the pathways? You sort of mentioned part of the pathways were affected. Can you shed some lights on how they might be affected?
Well, in fact, we looked at all types of variants, which means that they could be exonic, intronic variants, they could be intergenic variants, and we selected them. We selected the genes that might be implicated in the different types of neurofibromas, depending on the role of these variants that were already known. In fact, for example, we looked at EQTLs that were already known in some tissues that were already studied in the past. So we suppose that most of these variants are intergenic variants, so they do not have a direct impact on the proteins, but we suppose that they are modulators of the expression of the neighboring genes, and probably that's what's happening here.
Some additional questions. Great talk. Have you thought of how to evaluate the biodistribution? Oh, sorry, actually that was from the last. Somebody mentioned a very interesting study. Was there any adjustment or accounting for the phenotype severity, such as numbers of neurofibromas in your study?
In our study, we looked at the number of neurofibromas, indeed. So I'm not sure I completely understand the question, but we are looking for variants that are correlated to a higher number of neurofibromas in our patients and not just responsible for the generation of neurofibromas. So we suspect that these variants are correlated to a higher number of neurofibromas in the patients.
Okay. There are some additional questions. I might just selectively ask some of them. Someone asked, do you have plans to look for the presence of larger scale copy number alterations with the sequence data you have?
I'm not sure. Can you repeat? I didn't hear well.
Do you have plans to look at the presence of larger scale copy number alterations in your data set?
Copy number? Well, maybe we didn't think about it yet, but yes, we have a lot of data on this cohort. Really, we can do a lot of things. So yes, I think we can do that.
Okay, and did you look at the polymorphisms identified from prior candidate gene studies? Did any of them replicate in your GWAS study?
Well, there were a lot of GWASs that were performed in other traits, not in neurofibromatosis type 1. But there was a recent study in the study of the emergence of cutaneous caffeolate spots in NF1 patients. So we looked at the regions that they found, and we have no common regions between these two. So there are probably different genes that are implicated in caffeolate spots and in neurofibromas. And well, the other studies are usually not in no link with neurofibromatosis type 1. So yes, we have common regions, but I'm not sure this is really relevant.
In the interest of time, there are some additional questions I'll email you privately. In the interest of time, we should move on to the next study. Very interesting study. Thank you.
Thank you very much.
Excellent. So next up, we have Dr. Jay Pundavela from Cincinnati Children's Hospital. And the title of this presentation is T cells are essential for plexiform neurofibroma growth.
Before I start, I'd like to thank the selection and organizing committee for giving me the opportunity to present in this year's CTF meeting. My name is Jay Pundavela, and I'm a postdoc in Professor Nancy Ratner's lab at the Cincinnati Children's Hospital. What I'll be presenting here today will hopefully demonstrate the role of T cells in promoting plexiform neurofibroma development. Neurofibromas are tumors of the peripheral nervous system in NF1 patients. Tumors develop on the skin or nerve trunks, otherwise known as plexiform neurofibromas. This is caused by the loss of NF1 tumor suppressor gene in the Schwann cell lineage. It is known that neurofibromal microenvironment composes of all different sorts of cells, including NF1 Schwann cells, nerves, immune cells, and a cocktail of pro-inflammatory cytokines and chemokines. Among the immune cells first thought to play a major role in promoting neurofibromas were mast cells. However, it was shown that targeting the cytokine-chemokine pathway involved in mast cell recruitment did not prevent neurofibroma formation. Previous work in the rat lab has shown that macrophages have anti- and pro-tumorigenic function dependent on the stages of neurofibroma development. A follow-up study showed that by inhibiting the STAT3 pathway, reduced accumulation and proliferation of macrophages, which then attenuated neurofibroma in mice. Recently, we've shown that intervention of the CXCR3 receptor localized to T cells and dendritic cells prevented neurofibroma development. Based on all the current evidence, we proposed a model as shown in this cartoon that after the loss of NF1 in Schwann cells, mast cells and macrophages are recruited to the nerve, then followed by the infiltration of T cells and dendritic cells that drive neurofibroma initiation and macrophage retention, and ultimately a tumor growth mediated by cytokine and chemokine signaling. My research focuses on investigating whether T cells are necessary for neurofibroma development. So just briefly, T cells are immune cells known to play a key role in the tumor microenvironment. TCRs are receptor alpha and beta complex on T cell surface, responsible for recognizing antigen bound to MHC molecules on antigen-presenting cells or APCs. The ability of T cells to respond depends on a large repertoire of unique TCRs. This diversity of TCR is a result from somatic recombination events of VDJ gene segments during T cell development. This recombination event is mediated by recombination activation gene enzymes known as RAG1 and RAG2. Also, TCR diversity and its effector response is shaped by its interaction with the MHC complex on APCs. To explore the many phases of T cell in neurofibromas, by immunohistochemistry we found noticeable number of TCR beta T cells in all 5 tissue samples of human plexiform neurofibromas, whereas these are absent in a normal human nerve, suggesting that T cells in neurofibromas have the ability to engage with MHCs on antigen-presenting cells that would elicit an immune response. Then we wanted to look for the presence of TCR beta T cells in our NF1 flux DHH crew neurofibroma model, from here on named as NF1 mice, that mimics the pathology of a human plexiform neurofibromas. Spectral flow cytometry revealed an abundance of TCR beta positive CD8 T cells and CD4 T cells in tumors of the NF1 mice compared to a dorsal root ganglion of wild-type age matched mice. Gating out from these populations revealed the presence of effector memory T cells that are proportionally activated, indicated by the CD44 high and CD69 high expression. To test whether T cells play an important role in neurofibroma formation, we've crossed our NF1 mice with the RAG1 mutant mice. RAG1 mutant mice lack a mature T and B cells but have intact macrophages and other immune cells. Cross-dissections were performed on a 7-month-old cohort and shows that in the absence of T cells and perhaps B cells, neurofibroma formation is prevented. Also, a decreased trend in infiltrating mast cells were observed, but showed no significance. On the other hand, macrophages were significantly reduced in T cell deficient NF1 mice. It is worth noting that neurofibromas are characterized by dissociation of Schwann cells from nerve remote bundles. This is visible under an electron microscopy. To examine if this aspect of neurofibroma can be rescued in the absence of T cells, electron micrographs of the saphenous nerve from RAG1 mutant NF1 mice showed no improvement in the ensheathment defect of Schwann cells in NF1 mice, suggesting that Schwann cell nerve interaction is perhaps independent of T cell presence. So to test if T cells alone can directly promote neurofibroma development in the RAG1 mutant bearing mice or the T cell deficient mice, we performed a T cell adoptive transfer. Showing here in our workflow of the experiment, first spleens from donor NF1 mice were isolated and dissociated. Splenocyte suspension were then subjected to a negative selection in order to collect pan T cells, CD4, CD8 T cells, which are then to be used for the adoptive transfer. Meanwhile, blood were drawn out from a 6 weeks old recipient T cell deficient mice to determine the presence of T cells in the peripheral blood. After 7 days, adoptive T cell transfer by IP injection were performed to recipient T cell deficient NF1 mice. Blood samples are then collected every month post-injection to determine if T cells were successfully engrafted before they're taken for an MRI to assess the development of tumors. Recipient mice are then sacrificed at 7 months old for further analysis. Flow cytometric analysis of the blood from the T cell deficient NF1 mice pre-injection of T cells showed absence of CD3, CD4, and CD3, CD8 T cells in the blood. In contrast, abundance of blood CD3, CD8s and CD3, CD4 T cells is observed in the age-matched NF1 mice. Now, 1 month after the T cell adoptive transfer into the RAG1 mutant NF1 mice, population of CD3, CD4, and CD3, CD8 T cells appears to persist in the peripheral blood, suggesting that, demonstrating that we've had an efficacy of our T cell engraftment. To examine whether adoptive transfer of T cells will promote neurofibroma formation, MRI was acquired to the recipient RAG1 mutant NF1 mice. MRI images of a spinal nerve of RAG1 mutant NF1 mice that receive a pan T cell revealed the presence of paraspinal tumors. In contrast, no lesions were observed in a RAG1 mutant NF1 mice that received a PBS injection alone. Lesions of RAG1 mutant NF1 mice that received pan T cells were comparably similar to an age-matched NF1 mice. These data suggest that T cells promoted neurofibroma development. To determine which of these T cell subtypes play a tumor-promoting role, adoptive T cell transfer of CD8s or CD4 T cells were performed. MRI of RAG1 mutant NF1 mice that received CD8 T cells showed small paraspinal tumors, whereas CD4 T cell recipients didn't show a similar effect, suggesting that CD8 T cells may be the T cell subtype that promote neurofibroma formation. And so just to sum up, what I've talked about here today, TCR beta positive T cells infiltrate human and mouse plexiform neurofibromas. Among these T cells present in mice are activated effector memory T cell positive T cells. Absence of T cells prevented neurofibroma development, and when T cells were engrafted, neurofibromas were formed. And CD8 T cells are potentially the T cell subtypes that have tumor-promoting function in plexiform neurofibromas. And therefore, in conclusion, hopefully I've shown here that T cells are sufficient and necessary for plexiform neurofibroma development. Just to finish off, I'd like to acknowledge members of the Ratner and Hildeman Lab who contributed to the data presented here. Also, I'd like to acknowledge the funding bodies that makes this work possible. Thank you.
That was a very nice talk. I think we have a few minutes for questions. Waiting for things to come in. Have you thought about what this could mean sort of therapeutically for patients?
Yes, good question. So at the moment, we wanted to go further and try to look for what might be the sort of interaction that's responsible for like the MHC and TCR communication. So hopefully the ultimate goal here is we're trying to figure out what might be the antigen that might play a role in this. So if we could target that as well. Nonetheless, there are also anti-CD4 and CD8 blocking antibodies that are currently out in clinical trials in a different disease contexts. But how that could be used in NF is something that yet to be examined.
That's one more question from the audience. Someone had asked, did you observe any progression to MPNST in the mice with transferred T cells?
Good question. No, we didn't see any evidence of an MPNST. What's striking is some mice were getting skin diseases, but we were still trying to look into what sort of skin problems some of these mice were getting. There could be some sort of cutaneous neurofibromas, possibly, but this is just something that we could speculate on.
Okay. Good. Well, again, great talk. In the interest of time, we will move on. So I will turn it over to Dr. Chi to introduce the next speaker.
So now the next speaker is Dr. Lu Le from UT Southwestern, and the title of his talk is NF1 loss impairs the Schwann cell lineage differentiation to promote neurofibroma development. Welcome, Lu.
Thank you so much, and hello everyone. Can you hear me?
Yes.
Okay, great. Well, first I want to thank Pin and Angie and the NF meeting chairs for the invitation to share our work with you today. My goal in the next 15 to 20 minutes or so is to share with you the in vivo neurofibromogenesis assay that we developed and how we can use it to generate humanized neurofibroma carrying the same genetic mutation as in NF1 patient, so that we can learn its biology. And toward the end of the talk, I want to share with you unpublished data on how that help us discover new biology that regulate neurofibroma development. Let me see. Okay, let me see if I can move the slide. All right. So as you know, cutaneous and plexiform neurofibroma are tumors that contain the Schwann-like cell that originate from the neural crest. We are very interested in understand the developmental origin of neurofibroma, specifically the cells of origin that give rise to both the cutaneous and plexiform neurofibroma. So over the year, our lab and many other labs around the world have utilized different Schwann cell Cree to knock down NF1 at different state of their development. Some of these Cree led to only plexiform neurofibroma development. Some led to only cutaneous, and some led to both. So our lab using genetic labeling for cell tracing as a mean to go back and identify the very cell that give rise to both cutaneous and plexiform neurofibroma. And we was able to trace to the embryonic Schwann cell precursor in the dorsal nerve root, expressing both HOXB7 and SOC10 as one of the cells of origin that can give rise to both cutaneous and plexiform neurofibroma. one of the reasons that we are so interested in the identity of the neurofibroma cells of origin is because we argue that if we know where they come from, and if we have the ability to isolate this cell, then we can reconstruct all the biological steps from the cells of origin all the way to neurofibroma, so that we can help, so that can help us identify the rate limiting step or the Achilles heel of this process, so that we can target it to prevent neurofibroma from developing. So to do that, we collect the dorsal nerve root and DRG from the E13.5 embryo to generate the dorsal nerve root neurosphere cell, which we know contain the cells of origin for neurofibroma. We then ex vivally knock down NF1 and label them with LAC-Z and luciferase before we implant it back into the sciatic nerve of the recipient mice. And these mice would develop plexiform neurofibroma at the sciatic nerve that have almost the same molecular and histology feature as in a patient with NF1. Now, one of the advantages of this model here is that the tumorigenic cells are labeled with luciferase. So we can actually give them different pharmacological intervention, and then we can perform bioluminescent scan of these mice to objectively quantify tumor response to different pharmacological intervention. So here's an example that we did when MEK inhibitor became available. This is we did this 8 or 9 years ago, where we used the same assay. Tumor from a cohort of mice treated with either vehicle or MEK inhibitor, using the same in vivo neurofibromagenesis assay and using the bioluminescent scan to quantify tumor response. What we observed back then when we did this experiment is that tumor from a cohort of mice treated with vehicle continued to grow. But MEK inhibitor restrained neurofibroma growth. And after about 4 weeks of treatment, what we did next was we divided each of the group into 2 subgroups. One, we took off the MEK inhibitor, and one we continue with the treatment. And it's clearly shown that the tumor grow back after MEK inhibitor withdrawal, indication the need for sustained MEK inhibitor to prevent tumor growth. And that's exactly what happened when Brigade and her group did the human clinical trial a few years later, and her group saw similar observation that the plexiform neurofibroma grew back after MEK inhibitor withdrawal. So this really validates our in vivo neurofibromagenesis assay as a robust, fast, and a very cheap way to really perform in vivo drug screening for neurofibroma. So to make it more clinically relevant, we next decided to generate humanized neurofibroma model using the same assay to study the evolution from stem cell all the way to neurofibroma development. And to do that, we collaborate with David Goodman's group, who have generated a series of isogenic wild-type, heterozygous, and NF1 knockdown iPS cell that have the same genetic mutation as in NF1 patient. And these iPS cells basically carry all the marker of a true iPS cell. We would took these cells and implanted into nude mice and they would generate a teratoma, confirm that these are truly iPS cells. The next thing that we did was we was able to differentiate these iPS cells into Schwann cell, into neurograph stem cell, and then into Schwann cell linear cell, including the Schwann cell precursor that we observed that express a high level of SOC10 and HOXB7, the same kind of marker that we saw in the mouse neurofibroma cells of origin that were knocked out of NF1, give rise to both cutaneous and neurofibroma. So when we took these differentiated Schwann cell precursor from human iPS cell that carry the same NF1 mutation as in patient. And when we implant that into the sciatic nerve of a recipient mice, these mice develop a classic plexiform neurofibroma that carry almost the same molecular and histological feature and as in plexiform neurofibroma in patient with NF1. And it's as is expected, when we further knock out P53 in these iPS cell and inject them into the sciatic nerve of these of the recipient mice, they develop classic MPNST as well, just like in individual with NF1. Now, the most interesting thing that we learned as we differentiate the iPS cells into the humanized neurofibroma is that compared with the wild-type control, NF1 lost delay Schwann cell differentiation by maintaining them in the stemness state, where they more readily form a neurosphere, express higher percentage of stem cell marker compared to the differentiating Schwann cell marker, both in the human iPS cell as well as in the mouse DRG neurosphere cell. So most interestingly, when we carefully analyze the differentiating cell for the Schwann cell marker, what we found is that the delay in Schwann cell differentiation caused by NF1 loss resulted in an increase of Schwann cell precursor expressed in both SOC10 and HOXB7 in the differentiated Schwann cell precursor from the iPS cell. Now, this suggests that NF1 loss in Schwann cell lineage increased the pool of neurofibroma cells of origin by delay Schwann cell differentiation during development. So we believe that loss of NF1 not only led to the activation of the RAS MAP kinase pathway to increase proliferation for tumor formation, but it also delays Schwann cell differentiation to increase the pool of neurofibroma cells of origin that can help promote neurofibroma development. So the next question that we ask is, what is the mechanism behind this delay in Schwann cell differentiation? So to make a long story short, it is complicated, more granular rather than linear, but it turned out we found that SOC2 play a very important role in this process, where loss of NF1 in the Schwann cell lineage is associated with a very high level and sustained SOC2 expression. And this elevation in SOC2 expression is downstream of the MAP kinase pathway. We know that because when we treated these cells, NF1 knocked out that have a high level of SOC2 with a MEK inhibitor, it was able to rescue or suppress its elevation in the NF1 knockdown swan cell precursor. Now, normally in a normal physiology condition, SOC2 turn out within the swan cell lineage express highly during embryonic development in the swan cell precursor. But over time, as the swan cell differentiated into immature and more mature swan cell, the level of SOC2 dramatically drop. And that's what we see here on the upper panel here. When we take a normal sciatic nerve and when we stand for SOC2, both at the cross-sectional or longitudinal section, you can see that there's not a lot of SOC2 positive cell. But when we knock down NF1 in the swan cell lineage, we observe a persistent high number of SOC2 expression, both in the peripheral nerve as well as in both plexiform and cutaneous neurofibroma. This is not only in mouse tissue and mouse plexiform and cutaneous neurofibroma, but also in human tumor as well. So most importantly, when we use small hairpin to knock down SOC2 in NF1 minus swan cell precursor, and then differentiate them into more mature swan cell, what we observed was that they quickly lose their stem cell marker and express more differentiated swan cell marker compared to the scramble control. However, knockdown of SOC2 did not affect cell growth via this ATP cell titer growth assay that we did. So these suggest that SOC2 controls swan cell differentiation, but not proliferation of these swan cell precursor cells. So lastly, to determine the role of SOC2 in neurofibroma development, we went back to our in vivo neurofibromagenesis assay, where we knocked out SOC2 in the NF1 minus luciferase labeled swan cell precursor, and then we would implant them into the right sciatic nerve and the scramble control in the left sciatic nerve. And then we would perform a bioluminescence scan to quantify tumor growth. And what we found is quite striking. If you go from top down, these are the same animal. If you go from right to left, that's a different animal. But if you go from top down, these are the same animal that being undergo bioluminescence scan every week. And then what we saw in these mice is that while the tumor on the left sciatic nerve continued to grow, a knockdown of SOC2 prevent and severely delay neurofibroma formation in the sciatic nerve. So in summary, all of these experiments that I present today indicate that loss of NF1 in the swan cell lineage not only increase swan cell growth via activation of the MAP kinase pathway, but also delay swan cell differentiation to increase the pool of tumor cells of origin, make it more permissive to help promote neurofibroma development. And this process is in part regulated by SOC2, and the knockdown of SOC2 reverse this process and greatly impair neurofibroma development. So with that, I am going to stop here and acknowledge all the people who have funded the research in our lab, as well as the incredible contribution from past and current member of our lab, specifically John Moore, who did most of the work that I present today. I also want to thank our collaborators, especially David Gutman and Corina for this project. Thank you again, and I would be happy to answer any question.
That was a great talk, a very powerful system, Lou. I think we're going to wait for the questions to trickle in from Slido. In the meantime, so is SOX2 sufficient to sort of block dedifferentiation? Have you expressed SOX or get a chance to express SOX2 in the Schwann progenitor to see whether it can just by itself do that?
Yeah, so we are doing that now. Not only we overexpress SOC2, but we also overexpress SOC2 in vivo in the mouse as well. Obviously, it doesn't completely block the differentiation when we overexpress SOC2, but it severely delay their differentiation, and there's that not only just persistent delay, but eventually, just like differentiation, there might be delay. But if you look very late in life, then everybody will get there. This is almost like you run a marathon. Eventually, everybody get to the goal. But who get there first and how delayed they are really make a big difference, because these tumors, especially plexiform neurofibroma, there's only a window of opportunity where they are more permissive to form these tumors. And sometimes if you miss that window or if you fall within that window, it can dramatically either increase the chance or prevent these tumors from developing.
And then the other question I have is, what maintains the SOX2 expression? Do you have some thoughts on that?
Yes, that is something that we work very hard in that. We know that the downstream of MAP kinase pathway, because using MEK inhibitor, we was able to rescue that, and we they also not only just downstream, but they do have physical interaction with phosphor when we do co-IP also. So I think I wish the story is very linear, but I think there's just multiple ways. There's multiple regulation both at the transcription factor and at the protein level to maintain SOC expression to really alter or impair the Schwann cell lineage differentiation.
Great, thank you. I don't see any. There is usually a delay of questions. I think we still have a couple of minutes. Do you ever see, so CDKN2A is frequently lost in human plexiform neurofibromas. Do you see CDKN2A be loss or activation of the cyclin-dependent pathways?
Yeah, so we haven't looked at that because at least in our system we knocked out NF1 and we use it in a very massive amount number of cells of origin, because we use the tree that is very robust. So there is a driver. There's a reason why they develop a tumor. So as long as they are in a permissive environment where we put them right next to the nerve with NF1 and activation of the MAP kinase, and that's enough. They will push through plexiform neurofibroma. But then again, that is one of the assays. It might not be the most physiological assay. So under certain circumstances where the driver might be as strong, then you need other in the tumor microenvironment, including immune cells, fibroblasts, the heterozygous microenvironment. So I think it's complicated in a way that each of these models might be helpful to answer a certain question, but it might not be the model that we utilize for almost everything that we want to know about how these tumors develop.
Sure, it actually connects to the question that comes up. One person asked, it seems that the role of the immune system is not needed for this model to generate neurofibromas. Could this approach be translated to an immunocompetent model?
Yeah. So great question. Again, the way I see it is just like what I is flat, where these nude mice lack mature CD4 and CD8, but they do have uneducated T cell. They also have an intact macrophage, mast cells, as well as many other cells. And I believe it is not physiological in a way that we just put too much number of cells of origin there and right next to the nerve. And that's enough to drive these tumor formation. And clinically, histologically, they behave almost just like human plexiform neurofibroma.
One last question. Are other SOX factors or genes, transcription factors involved in Schwann cell differentiation and neurofibroma formation?
Yeah, great question. And we did look at that. For example, SOC10. SOC10 is just a vital transcription factor for Schwann cell differentiation. We did not see SOC10 upregulation up in NF1 knockdown. So not all SOC function the same in Schwann cell differentiation. Some are vitally required, but not regulated by NF1 loss.
Thank you. I'm going to hand the podium back to Angie Herbie for the last talk of this session.
Thank you.
All right. It is my pleasure to introduce Dr. Liam Laraba from the University of Plymouth in the UK. The title of this presentation is NF2-TEAD autopomidylation inhibitors slow tumor cell proliferation in the preclinical periosteal schwannoma in vivo model.
Hello everyone, my name is Liam Laraba. Firstly, I'd just like to thank the CTF for giving me the opportunity to present my research on NF2 null schwannoma today. And I'll be talking to you all about TEAD palmitoylation inhibitors slowing tumor growth in the preclinical periosteal schwannoma in vivo model. So just an overview first of what we're talking about today. Firstly, I'll give you some background on Hippo pathway activity in the context of NF2 null schwannoma. I'll talk to you about the therapeutic potential of TEAD inhibition, the study design that we used, and in particular the periosteal NF2 flox mouse model. Then I'll show you that TEAD inhibition reduces proliferation of primary human schwannoma cells in vitro, as well as mouse schwannoma tumors in vivo. And then I'll summarize everything at the end. So the Hippo pathway is an evolutionarily conserved pathway that controls organ and tissue size, and it centers around this core kinase cascade whereby MST phosphorylates LATs, which phosphorylates YAP and TAZ. And when YAP and TAZ are phosphorylated, they are retained in the cytoplasm by 14-free protein and subsequently degraded. And the NF2 gene product Merlin is critical in transducing signals from the cell membrane, as well as providing a scaffold to facilitate this core kinase cascade. So in NF2 null schwannoma, in the absence of Merlin, you don't get this cascade, which means YAP and TAZ remain unphosphorylated, whereby they translocate into the nucleus and activate transcription of various transcription factors, with the best characterized of these being perhaps the TEAD family. And TEAD transcription then leads to tumor phenotypes. So in all 4 TEAD family members, there's a conserved cysteine residue within the YAP-TAZ binding domain, which is palmitoylated. And this palmitylation is shown to be crucial for YAP-TAZ binding and therefore YAP-TAZ-driven TEAD transcription. Which is why we were very excited at the CTF meeting in Paris 2018, where Vivace Therapeutics approached us to talk to us about their drugs, which I'll refer to as VTs from hereafter, that really potently block this cysteine palmitylation in the YAP-TAZ binding domain, which means you don't get YAP-TAZ binding and you don't get YAP-TAZ-driven TEAD transcription. There is also some evidence to show that blocking palmitylation can lead to reduced TEAD stability and possible TEAD protein degradation, and this is something that we also looked into. And recently our collaborators with Vivace have published an interesting study in which they show in NF2 deficient mesothelioma that they can use VT drugs to really strongly block proliferation at clinically relevant nanomolar concentrations. So we were really excited to try out their drugs in schwannoma. So firstly, we did some preliminary data in human schwannoma, and now we are currently testing out their drugs at scale in NF2 null mouse models. So this preliminary data, firstly, we just wanted to look in NF2 null human cultured schwannoma cells, and we wanted to just check for TEAD expression in 3 different schwannoma lysates to check if we could use an isoform-specific TEAD inhibitor if there was a dominant isoform in schwannoma. However, it turned out to be a really, really mixed bag. So we went with a pan-TEAD VT inhibitor, and treatment with this inhibitor significantly downregulated the expression of the hippo readout gene, CTGF, or connective tissue growth factor. So this is a TEAD-driven protein that when we give the VT drug is strongly downregulated, as you can see here. But what does this actually mean for proliferation? Well, after 48 hours of VT treatment, we performed an EDU proliferation assay where EDU positive cells are labeled red. And as you can see, as we increase concentration, we get a strong reduction in proliferation, which is quantified here. And you can see the IC50 came out at 39 nanomolar. So this provided us with a strong mandate to take our studies into a schwannoma mouse model. And the model that we used was the Periostin-Cree NF2 flox mouse model, published by Gelhausen et al. In 2015, part of Wade Clapp's lab. And in their model, you can see that you get spontaneous tumor formation in the dorsal root ganglia, or DRGs, and vestibular ganglia, or VGs, as I'll refer to them from hereafter. So you can see the tumors here in the dissected spinal cords of Cree-positive mice, and also the Schwann cell hyperplasia in these H&E images of the DRGs and VGs. Now, this model is a Schwann cell specific deletion of Merlin, by which they use the periostin promoter, which is Schwann cell specific, to drive the expression of Cree recombinase. Cree recombinase then excises the NF2 gene, which has been flanked by LOXP sites, which results in a Schwann cell specific deletion of NF2 and the formation of these spontaneous schwannomas. And we combined this model with our YAP flox and TAZ flox knockout mice to generate NF2 YAP double knockouts and NF2 TAZ double knockouts in order to compare genetic ablation of YAP and TAZ to pharmacological inhibition of the VT drugs. Our experimental workflow was this. So we took 3, 5, and 9 months to sort of model early, intermediate, and high tumor burden in these mice, and you can see by H&E images, we can see Schwann cell hyperplasia in Cree-positive mice at all 3 of these time points. We tested out 2 drugs, VT1 and VT2 from Vivace, that showed good oral bioavailability and safety in mice, and we sort of gave VT1 at 10 milligrams per kilo and VT2 at 30 milligrams per kilo by oral gavage for 21 days, following up with a 24-hour EDU pulse to track proliferation at the 2 tumor sites, and also we took the sciatic nerves to be able to Western blot for Schwann cells and look at protein expression. There's various immunofluorescence approaches that we can use. We can use a PLP-GFP transgene, which allows us to tag Schwann cells with GFP. We can use an S100 stain to visualize Schwann cells as well, and we can use a neurofilament antibody to depict the architecture of the VGs and DRGs, and of course EDU to show proliferating cells. So in our genetic data, we show here that in Cree-positive single knockout NF2 null mice, you get a significant increase in proliferation in both DRGs and VGs compared to Cree-negative controls, quantified over here. And when we remove either YAP or TAZ, we get a partial rescue of this elevated proliferation in both DRGs and VGs again. So then this led us on to the pharmacological inhibition of hippo signaling. So I'll show you VT1, which was 10 milligrams per kilo again at 3 months in the DRGs. And you can see here high amounts of proliferation in the Cree-positive vehicle. But when we give drug, we get a quite large reduction, significant reduction of about 50%. And you can see when we Western blot the sciatic nerve that CTGF is strongly downregulated in Cree-positive drug-treated mice, which is quantified down here. We didn't see a change in TEAD expression with VT1. VT2, 30 milligrams per kilogram, this time in 5-month-old mice, just to show you our variety of data. Again, Cree-positive vehicle mice proliferating, and then much decreased in the Cree-positive drug mice, about 50% again. Similarly, CTGF expression significantly reduced. This time, we also saw a significant reduction in TEAD expression with the higher dose drug, showing target engagement again, with reduced TEAD stability. In the vestibular ganglia, we've tested out both drugs just in the 3-month time points at the moment. So VT1, you can see here, elevated proliferation in the Cree-positive mice, and then when we treat with drug, back down, this time perhaps more efficacious with a reduction of around 72% when we quantify. And then with VT2, reduction around 50%, but again, still efficacious Cre-positive vehicle versus Cre-positive drug. So with that, I hope I've managed to show you that the VT inhibitors that we're using significantly reduce NF2 null schwannoma proliferation in vivo. It's really important to note that mice show no significant weight loss or any other adverse effects during the 21-day gavage period, and we're really looking forward to taking our cohorts of 9-month-old mice through these treatment regimes to see the effects of these drugs when the tumour burden's high. I hope I've managed to demonstrate to you that mechanistically VT inhibitors are repressing T transcriptional activity. We can see this by the downregulation of CTGF in the Cre-positive drug-treated mice, and both drugs seem to be having a pretty equal efficacy in terms of reducing the proliferation. And then just to add, there was some quite exciting news from Vivace, who said that they've managed to get one of the VT drugs into a clinical trial, a phase 1 trial. So we're really hoping that this can accelerate the translation of our model in schwannoma into the clinic as well with these drugs. So with that, I'd just like to acknowledge our lab, the lab of Professor David Parkinson, our in vivo facility technicians, the Hahnemann lab, who also provide great support for us, our collaborators Vivace Therapeutics, Dr. Tracy Tang and Dr. Len Post, and of course our funders Vivace, who provide support and the drugs, the CTF for funding my Young Investigator Award, and then I'd lastly like to thank all of you for your attention, and I welcome any questions.
Very nice talk. We have just about a minute left, so we can see what questions we have. So one question that someone has is, do you have plans to test on meningiomas or ependymomas?
Yes, so at the moment we have, we're just doing pilot studies with orthotopic xenografts with KT21 and BEMM1, which are two Merlin null meningioma cell lines, sort of low grade and high grade, and we can do these orthotopic xenografts into skid mice, and then pretty much do a shorter treatment regime, and then see the effect of meningiomas. Yeah, that's something that's underway.
Someone wanted to know what disease type is being tested in the clinical model, the clinical trial that they have begun, that Vivace's been able to start.
So it's phase 1, but I'm pretty sure that it'll be following up the NF2 deficient mesothelioma work that was recently published, that was in my presentation.
Okay. And another question, what would be needed to bring these drugs to NF2 patients?
I suppose the success of the phase 1 trial, and then just completing the cohorts in 9 months and seeing the efficacy in our in vivo model to then move into clinical trials, and also as somebody mentioned, the concomitant work with the meningioma, because with it being NF2, we can sort of target both pathways, both tumor types, hopefully.
All right. Again, that was a great talk. I'll turn it back over to Ping to wrap things up.
All right. We would like to thank all of the speakers and participants for a stimulating session, and this concludes the tumorigenesis session. We hope to see you in other sessions and in person next year. Thank you.